development of the turtle limb. And so here we have a turtle, and this turtle, stage 15, and it looks, it's a male turtle. You can tell it's a male turtle. And we grow our turtles at male temperatures. And you have the cartilage and the muscles, and we have the limb bud here with the AER at the tip. And sure enough, we find, when we clone what we think is turtle FGF10 gene, and this was done by degenerate primers, we couldn't be certain it was, but it had the same expression as chick and mouse FGF10, namely, it was found in the genital tubercle, it was found in the muscles of the limbs, and it was found in the mesenchyme adjacent to the AER. So it had sequenced 96% homology to the FGF10, 90% amino acid homology to FGF10, and it was found in the same place as FGF10. So we thought, okay, great, we can check that off. Just like in the limb bud, there is FGF10 in the underlying mesoderm. So if it's like the limb bud, we should expect FGF8 in the AER and in the, well, actually, the next slide here. We found FGF10 was also in the carapacial ridge, just underneath the ectoderm. So the mesoderm of the carapacial ridge also had FGF10, and here's the rib coming into the ridge, and here is synthesis of FGF10. Okay, so we can check that off. FGF10 is in the carapacial ridge. Let's look for FGF8. So here's here's where we would expect to find it. Here is the carapacial ridge. We'd expect to find it in the ectoderm of the ridge. And here's the apical ectodermal ridge, our positive control for the chick. You know, we know that that FGF8 is found in the AER, and sure enough, it's found in the AER, no problem. And when we went to the ridge, we could not honestly say that we got it. We tried different ages, different hybridization techniques. No, FGF8 was not in the carapacial ridge. Okay, let's take a step back. Let's work on the classroom. So we're not going to work on this for a while. A student came into our laboratory and said that she wanted to work on the question of turtle development. Is there space in the lab? We said, sure. Why don't we teach you how to do in situ hybridizations? So we taught her how to do in situ hybridizations. She then said that she's ready to try one by herself. Fine. Here's the probe for FGF8. We have plenty of FGF8 probe. We have these embryos which were too old for us to use. Show us where FGF8 is. And we expected it to be in the brain. We expected it to be in the AER. And she comes back, and unfortunately this is a rather dark slide today, but she found it in the tips of the rib. The tips of the rib have FGF8. And this makes it like the limb bud. There can be an interaction between FGF8 and FGF10 to expand the rib and the ridge together. So it looked like there was an induction here between FGF8 and FGF10. Can we show this? Well, turtles allow us to do things in culture that we could never do with chicks and mice. Turtles love anoxia. At conditions of culture which would destroy chicks and mice, the turtles survive. The head of our animal rights committee, when we gave our protocols to her, said, why don't you kill turtles by putting them in CO2 tanks? And so I said, we don't have 100 years. You know, we have to do this experiment this week. It'll take them months, years to die of anoxia. So, we grew these in culture. What you see here are turtle embryos where they've been, the bottom part, the viscera has been taken off, it's been cut away. So these are dorsal turtle embryos. They are put on filter paper and then floated on culture medium, and they can be kept over a week. Matter of fact, their tails start moving. It's really bizarre. But in control media, we get an AER format. right over here. You can see the AER. It actually comes above the plane. You know, the turtles are pretty flat, and now it's rising up. Is this apical ectodermal? Is this carapacial ridge? But if we add an FGF inhibitor, that ridge disappears. See that by this blood clot is where the ridge was. And what you see are the ribs are going ventrally. You can see the ribs turning and going underneath. They also, as a positive control, have a poor AER. Okay, so here's our internal control, is the inhibitor working. Yes, the inhibitor is working, and here we get a nice carapacial ridge. Here the carapacial ridge degenerates and the ribs go ventrally. When we did the converse experiment with chicks, we could actually get the chick rib to go to a place of FGF10 embryos. So in normal, when we just use saline beads on our, when we just have beads coated with saline, the ribs didn't care about it at all, and they just migrated as chick ribs usually do. They migrated ventrally. However, when we had FGF10 on the beads, the ribs actually came towards the beads. And so we think that there is migration of the ribs due to a new source of FGF10 being produced in the carapacial ridge. And this leads to these wonderful morphologies, axial displacement of the ribs in hard-shell cryptodeer and pleurodeer turtles, where the ridge, here's the carapacial ridge, and the ribs and the ridge grow together, probably through this FGF10, FGF8 interaction. And you can see they start off as cartilage. They later become bone. And we'll talk about that in a moment. In soft-shell turtles, studied by Shigeru Kuretani, you get the start of the carapacial ridge, but then the carapacial ridge outgrows the ribs, and so the ribs only form a small plate in the turtle embryo. So we have this wonderful outgrowth of the ridge and the rib. We also, the ridge also is involved in integrating the scutes of the turtle, but I won't go into that right now. We can ask after that. So we are here. The ribs migrate into the carapacial ridges, probably because they are attracted to a source of FGF. FGF10 is definitely there. The outgrowth of the ribs and the carapacial ridge is probably coordinated by a positive feedback loop between the carapacial ridge FGF and the FGF8 on the ribs. So, next question is, what do the ribs do to actually make the shell? And what they do is what ribs are supposed to do. They undergo endochondral ossification. The cartilage is being made into bone. This is what normally happens in vertebrate ribs. You start off cartilage, and this is Alcyon blue showing the cartilage, and alizarin blue showing the cartilage, Alcyon showing the cartilage, and alizarin red showing the bone. And you can see that the ribs start off as cartilage, and they go to the periphery of the turtle embryo. Then what you see is they turn red. They turn into bone. And this is the same turtle. This is just different aspects of the same turtle. You see the blue over here, that's still cartilage. The bone starting to form in the center, working its way outward. This is the remodeling zone where the bone is becoming cartilage. And what you see here also, you can see on the ribs these spurs of red, which indicate that the cells around the rib are becoming bone. And when you look a little bit later, around the time of hatching, you see the nuchal bone over here, and then you see the vertebra, and you see the ribs, and around each rib there's a flare of new bone being formed from the dermis, and sometimes they're getting connected. By the time, about 2 months after hatching, you could see that the ribs are coming together to form the costal plate. The costal bones are forming, and they are going in between the ribs. So what we think is happening is that the ribs are going out as to the periphery, attracted by FGFs. And once they're there, they undergo endochondral ossification. But now they are surrounded by dermis tissue. And dermis, we know, can respond to bone-forming signals, especially BMPs. So one of the signals, one of the paracrine factors that is known to ossify both skin and cartilage is BMP4, bone morphogenetic protein 4. And we know that bone morphogenetic protein 4 is in the periosteum of the cartilage, and perichondrium of the cartilage and the periosteum of bone. So we wanted to know if dermal cells can respond to BMP by turning it into bone. Maybe what's happening is that each rib is acting as a signaling center, putting out BMP4, and when it puts out BMP4, not only can the cartilage respond to it and become bone, but the skin can respond to it and become bone. And one clue to this was the skeleton of Henry Eastlack. Henry Eastlack donated his skeleton to the Mutter Museum to study it. He had a disease called fibrodysplasia ossificans progressiva, the progressive ossification of his skin and connective tissue. It is a lethal disease. It is a disease that often kills people in their 30s. They suffer basically a paralysis where they cannot turn because their connective tissue has been turned into bone. And you see all these ectopic bones over here. The lymphocytes are involved because whenever a person has trauma and lymphocytes go there, the lymphocytes are not only making antibodies, the B cells are also making BMP4. And this was shown by Fred Kaplan and his colleagues at University of Pennsylvania Hospital, that there is a mutation which causes the lymphocytes to make BMP4. And so when they go to put out their antibodies, they are also putting out bone morphogenetic protein. So that was one clue that says we should look at BMP4. The other clue was just in the signature. Was here's the human BMP4 amino acid sequence. You really can't, you can't ignore these things. Okay. So it's unfortunate that this is focusing so darkly. But what we see here is this is the cartilage of the rib. Here is the bone forming around that cartilage, and you can see over here the dark compound. Now, what this dark compound is, it's not BMP4. We know that BMP4 is going to be made from ribs. What this dark compound is, it's phosphorylated SMAD1. That's one of those signal transduction molecules. It's the molecule that gets phosphorylated when BMP4 hits that signal. When BMP4 meets the receptor, it phosphorylates the receptor phosphorylates the protein SMAD1, which will act as a transcription factor. SMAD1 is phosphorylated, and this shows that the cells around the bone here are being phosphorylated. This is just a higher resolution of that. You can see the cells around the periosteum being phosphorylated by BMP4, and you might be able to see this little groove here. When you look at bone formation, a few slides later, you can see that groove is actually the beginnings of a bone. This is on, one side of a bone. That what was BMP4 reception, that the skin is able to receive BMP4 and turn itself into bone. This is just another picture of these cells. You can see they're being ordered. The cells here are expressing in their nuclei the phosphorylated SMAD1 protein. Here a bit further away, they are not. And they are being organized to form bone. So, Trachemys, the turtle costal bone, seems to be formed in two ways. One is this early periosteal expansion, which you can see over here. The bones are getting kind of bigger, broader. And then later, they are transforming the skin into bone. So here we have the alizarin red and blue. Here's the cartilage, here's the bone. And you can see over here, this kind of, this forming of the canals over here of the bone. And sure enough, when you use the phosphorylated SMAD1 antibody to ask, is the skin responding to BMPs? It's saying, yes, this is where the skin is responding to BMPs. This is where you're going to have bone forming. So, the model that we have for carapace formation, FGF10 is secreted by the carapacial ridge dermis. The ribs migrate into a source of FGF10. FGF10 is usually not made in the dermis. This is the new thing that helps create the turtle. That and the fact that it has a thick dermis that a rib can get into. The outgrowth of ribs in the carapacial ridge seems to be coordinated by a positive feedback loop between FGF10 and FGF8. What I haven't shown you is that Wnt and beta-catenin are involved in mediating these signals just as they are in limb buds. Each rib serves as a signaling center for the transformation of the costal bones of the carapacial plate and the growth of the bone from the rib periosteum outward. So when you look at a turtle shell skeleton, what you see is that it's connected to the bone. Each bone of the carapacial plate is connected to the rib, and it forms sutures where it meets that bone connected to another rib. In other words, the sutures are in between the ribs. A major signal for costal bone formation is a BMP, causing the dermis to ossify around the rib as the rib undergoes endochondral ossification. Okay, getting back to the creationists and their personal attack. Okay. The title of the paper by Leeson and Gilbert summed up the evolutionary conflict perfectly. Okay, I emphasize Leeson. Tyler Leeson was a student of mine. He worked on turtle shell development with me. He later goes to Yale University and becomes a turtle paleontologist. And what does he do? He actually finds the original turtle. He discovers the animal that was the proto-turtle. Did he have to go to South Africa to discover it? No. It was in a Yale Museum drawer. And he looked at this skeleton in a way nobody else did because he has new techniques. One of the things about a turtle is that it has no intercostal muscles. It doesn't have spare ribs. Okay, you can't eat the turtle ribs and get any meat from them because they didn't follow the myotome. They didn't follow the muscle-forming part of the embryo. They went into the dermis. There was no muscles for them to get to in the dermis. So, turtle ribs don't have intercostal muscle connections. The only ribs that do are the front and the back, where there's kind of the end at each kind of makes a basket with the muscles. So they looked at this fossil, Uninosaurus africanus, from 260 million years ago, and now they have the techniques, and they have enough of them so they can section these embryos, these fossils. They're not embryo fossils, they're adult fossils. And what they find is no intercostal muscles between the ribs. You can look at the ribs and see if there's attachments for the muscles. In most vertebrates you find them. In this one you do not find them. And this becomes the potential ancestor for the turtle. And the growth of the bone outward is seen from the rib periosteum, just as in turtles, the T-shaped broadening of the ribs, just as in turtles, no insertion points for the intercostal muscles, just like in turtles, the shortening of the trunk to 9 dorsal vertebrae. It looks like a primitive turtle. And Tyler made, I'm very happy he called this the evolutionary developmental model for the origin of the turtle shell. He could look at the ancestors and look to see that there are developmental stages that actually resemble, in general, some of these ancestors, but the most important ones are involve the lack of intercostal muscles and the paring down to the 9 vertebrae. So he was able to show that we now have a mechanism for the evolution of the turtle, and we have a potential ancestor for the turtle. When Tyler wrote that paper, 2013, there were 5 extant cladograms, depending on where you wanted to put the root, for turtle evolution, including one that he made erroneously a few years before. And he was able to show, yes, Here is the ancestor of the turtle, and at about the same time, genomics came out and said that the turtles are related, their sister group, to the archosaurs, which made a lot of sense with this model. Okay, so how did the turtles get their carapace? The carapace is formed by heterotopic bone formation, where a new function is found for the rib. FGF10 regulatory gene expression is found in new places. It's found in the carapacial ridge in the dermis. It makes this ridge. No other animal that we know of makes this ridge. The rib then migrates to new places, places where the rib has not gone before. And then what? And this is an FGF-mediated, fibroblast growth factor-mediated process. And then BMP mediates bone formation in a new place. So you get the formation of the shell. So what Huxley said, evolution is not a speculation but a fact. It takes place by changes in gene expression, basically, by epigenesis. To understand evolution, one needs to understand development and how development can be changed. And I'll end here with a quotation from David Plowman. Nature interests me because it is beautiful, complex, and robust. Evolutionary theory interests me because it explains why nature is beautiful, complex, and robust. Thank you very much. Shout out here to the various funders who made this possible. This was done almost totally with undergrads. I left out the part that was done with postdocs. This was done mainly with undergraduate students who worked in my laboratory. If you want, we could talk about fast drawing formation during the question and answers, but I don't think we can go into it right now. So, do you have any questions? And did I finish on time or did I go over?
No. Perfect is perfect. So there's all time for questions.
Okay, great.
And as I said before this morning, also questions your talk is not are allowed right now. Okay, so Silvia, yes, questions?
I have a question from your talk this morning.
Yes.
Because you talked about the interaction of the microbes and your biome, and I was thinking, at this point, what we are doing now for personalized medicine, with all these data cells, that become the different tissue, you put the human body on chip, you do everything in GMP way, then how we can also include all the microbes, right? You think we have to study all the community, or we have to study specifically each one what is doing? Because when I test a drug on a tissue, I think at this point I need to consider what the microbes around this tissue are doing to the drug.
Oh yes, this is, I think that personalized medicine, personalized pharmacogenetics has to be based on the holobionts, because different people, not only, first, different people have different microbes in them, and those microbes will digest digitalis differently, they will digest birth control things differently, they will digest cancer drugs differently. So you have to know what the microbiome is. You also have to know what their diet is, because the microbiome can change at the bite of a hamburger. When you have red wine, you allow certain microbes to proliferate, and white wine a different set. Because there are some bacteria which will take the tannins in red wine and use it for to metabolize. So our microbiome, I think, is critical, and this has been shown in tests in immunology, where they were trying to induce a particular phenotype in a mouse, and they always got the phenotype induced, a certain percentage of the time, when they injected this stuff into the mouse. And then they get a mouse from a different supplier. It's the same genetic strain of mouse. They feed it the same things. and they get a totally different result. They can't get the phenotype. And they find out the difference is in the segmented filamentous bacteria, which was present in one supplier, but not in the other supplier's mouse.
At this point, the idea is not to work in a completely clean situation, but to take in account as a variable of the...
Yes. I think it makes biology more difficult. Well, but it should make us suspicious of claims that, you know, the Human Genome Project is going to solve all of our pharmacogenetic problems.
I also have a question about your theory. So you mentioned that 90% of the serotonin is made by basically gut microbiome.
Right, 90% of the peripheral serotonin, not the brain serotonin.
Right. Based on that, and based on, let's say, all the epidemiological stuff that we are seeing these days in the last decades, so explosion in, diabetes, obesity, epidemics, depression epidemics, right? So my question, for instance, for depression epidemics, should we study the microbiome of hypertonic people and maybe get, use them for fecal transplant as a possible option?
Okay. If I had my slides from this morning, I would have. I have some slides of that data. It's starting to come out. Matter of fact, the same month that we lost the gene for depression, that the big study was done that says that the thing that we thought, the serotonin uptake receptor that we thought was the gene for depression, no, that's not the case. Another group came out saying, well, look at the microbes. We have a loss of a particular microbe in the fecal pellets, the feces of depressed people. And they link it actually to a certain bacteria with a certain metabolic pathway, which is producing a chemical which we might need hormonally, tyrosine, I think tyrosine or tryptophan derivative. Anyway, they've linked it to a pathway. Now, this is the first paper in this, and so I don't want to say this is anything definitive, but they have evidence that human depression may be caused by loss of some microbe. Similarly, a paper came out 2 months ago. It was a follow-up study of a very uncontrolled autism study looking at fecal transplants of children with severe autism, and showing that 5 years after the fecal transplants, giving feces from healthy people into people with syndromes, that they got a marked increase in sociability and in the lack of repetitive behaviors in these children who had the fecal transplants. Now, they did not have a control group. They had 18 people. This was a very small study. It was published in Nature Communications, I think. But it's important to say people are starting to do this. The correlation of different feces microbes with autism was done in the 70s. And so that was something that was on the books early on, but no one knew what to do with it, and no one thought the microbes were important. Martin Blaser at Columbia University has a book where he worries about the fact that we have this obsession with cleanliness. And he says that our obsession with cleanliness may have eliminated certain bacteria that we might need for maybe a small cognitive boost, or maybe a small, suppression of arrhythmia in our heart. And we, just like the zebrafish model, where Aeromonas is a very rare bacteria in the genome in the bacteriome of zebrafish. Maybe we need trace amounts of these microbes, and we're getting rid of them. It would be like getting rid of zinc in our body. So he is suggesting that we really study hard what the microbes are doing, and as Charles Knight said, eat more dirt. That we're just too sterile, and we're not made, we didn't exist. A friend of mine explains the current situation in politics throughout the world by just this phenomenon. That the only way to determine is these people are not getting the microbes they needed when they were kids.
already have one question on this group and one question on this one. So I'll start with this, so just to change it. So I know that you cannot give an answer to my question now for starters, but I want you to extrapolate from what I'm asking to other systems, because it's very nice and beautifully presented, the hypothesis, but then the expression in different domains of these two key signaling molecules, FGF and BMP, occurred in battle that could explain this, the evolution of the shell. Now, those genes have been shown to change, as you also showed in the introduction, in many other animals,
Yes.
also in the so-called model system where you can track all the modifications. So, is there any paradigm on the mutation of the cis-regulatory module of this gene that can suggest you how this can be evolved?
Yeah.
The second question together. So
No. Well, the answer is we do not know what genes are involved in making FGF10, for instance, turn on in the carapacial ridge or make a carapacial ridge. It looks like MEF2 is involved, but and so it's probably not so much a case of BMP being, having a difference, but maybe MEF2, which controls. And the turtle genome, there are two draft genomes. They aren't very complete at the moment, but we hope to look at these genomes to find out what they're doing, but we don't even have the right question. Yeah.
And so, going back to this morning, something I never thought about, but could be that, is there any studies concerning babies that were born by cesarean cut and were not breastfed from the mother?
Yes.
And to see if there is any increase in disease.
Yeah. This is the big question. And yes, the answer is yes, there are now several studies. And they differ as to where you are. What is known is that the babies who have not been breastfed and who were born by cesarean, especially those who were born prematurely, do have a different set of bacteria, they have a different set of immune cells, both B cells and T cells in their immune system. It's also known that babies who are born by cesarean and not breastfed have higher incidences of morbidity, hospital visits, and so forth, especially if they are in countries where bacterial diseases are endemic. Sub-Saharan Africa, for instance. This is going to be really deleterious to them. It's also known that in the West, you have slightly more, statistically more morbidity, not necessarily mortality, but morbidity, more hospital visits. But by around the first year, they kind of come together, that the microbiome settles into the same type of microbiome in both cases, those born by C-section, those born by birth canal, that they kind of converge at about a year of age. So it's that first year, and it's kind of like, if you can get through the first year, you're golden.
Always regarding the seminar lesson or this morning, you said that all metazoa are considered holobiont. So I'm wondering what is the first metazoa? When we can trace back the first?
Okay, excellent. Really wonderful question. So it's known that protozoa, protists, have bacteria, lots of them. Now, what's just fascinating, you mentioned metazoa, and it is very possible that the origin of metazoa was from a symbiosis of choanoflagellates with certain Bacteroides bacteria. Bacteroides us go way, way back. So there's a Bacteroides bacteria called Algoraphagus. And when you take choanoflagellates, which are the sister group to animals, sister group to the metazoans, and you put them into culture in distilled water, they stay monocellular. They stay unicellular animals. However, if you add back the Algoraphagus bacteria to them, or some lipids produced by the bacteria, the protists, the choanoflagellate protists, when they divide, will not separate. And they'll divide like 4 times. And they will have intercytoplasmic connections between the cells, and they will form an extracellular matrix like an epithelium. So it looks like multicellularity, at least on this level, can be induced by the symbionts. And there are 3 lipids that are involved in this. One produces the first division, one which extends it to other divisions, and one which inhibits it so that it goes so far and no further. And so we have a nice paradigm here for the origins of multicellularity through symbiosis. Now, there's another thing about choanoflagellates. They can also sometimes undergo meiosis and have sex. Usually they just, do mitotic divisions. But in the presence of a different bacteria, they will join together head to head, undergo meiosis, and transfer nuclei. So symbiosis may have been, may have been necessary for the origin of both sexual reproduction and multicellularity, 2 really big things. As you know, the caveat is the same caveat about mathematical modeling in microevolution, that what the answers you get can tell you what could have happened. They didn't tell you what did happen. They tell you what's possible. They don't tell you what it was. But working with extant animals is the only way we can do this experimentally. And this is work from Nicole King's lab at UC Berkeley. Really fascinating stuff.
Any more questions?
So we like to prosecute the rest of it. Thank you.